position breaks a woman's water, okay, or a woman says my water broke. We're talking about this bag of waters, which is the amniotic sac. So the embryo fetus is in this amniotic sac. If a woman's labor isn't progressing along well, it's too slow, and the amnion hasn't ruptured, the doctor can come up through the cervical canal and tear it. Essentially pinches it and tears a hole in it. And that usually causes labor to become more intensified. There's a problem if a woman's amniotic sac ruptures on its own and then labor doesn't progress. Infection can start if the baby hasn't been delivered within 24 hours. So they'll want to give Pitocin or oxytocin, a form of oxytocin, to cause contractions to start giving the mother antibiotics to make sure that there isn't an intrauterine infection. So with our question here, which of those structures would have to be opened to gain access to the amniotic cavity? So we had B, okay, decidua capsularis, chorion levi, and the amnion. Correct. If we go back to that original, not the original, but the previous image, so that instrument would be passing up through here. First layer to be reached is the decidua capsularis. The next layer is the chorion levi, and then the third layer gaining access to the space which contains the fetus is the amnion. Right? So in my diagram, here would be the decidua capsularis, chorion levi, and the amnion. Okay? All of those are the barrier between the fetus and the uterine cavity. So it's not just the amnion. When you reach up there and examine, you're not going to be touching the amnion. Those layers are thin, but those layers are there.
How hard is it to pass through that mitosis plug?
The plug? Not that difficult. If it's still there. It's often gone by then. All right, so here's some images of placentas without the babies. Somewhere out there in the world.
Why do people eat this?
It's a cultural thing. Animals will often eat their placentas. It's high in protein.
Why do humans eat it? We don't need the protein that bad.
No, there's hormones and protein. Other than that, I mean, you know, why do we do any of our traditions? Certainly, I was still in the morning class, my daughter this weekend working down at the horse farm. One of the horses was supposed to be pregnant. She went out to bring it in to feed, and the horse was lying down, and the farm dog was eating the placenta. And they looked over the entire area and could not find a foal anywhere. So usually the foal comes first and then the placenta, usually the baby and then the placenta. So whether it was born stillborn and the coyotes, I told her maybe the dog ate the foal too. So they weren't expecting for another 4 or 5 weeks, but still, I mean, we're talking relatively large foal. But so this surface would be the amniotic surface, this shiny gray material that you're seeing here is the amnion. And we turn it over, the bloody surface is they're called cotyledons. It's the stratum functionalis. So what we're looking at there on the maternal surface is right here. This area right here, where the stratum functionalis separated from the stratum basalis. So we're looking at the deepest part of the stratum functionalis, and is the decidua basalis. So in this view, we had twins over here with 2 umbilical cords. And again, maternal surface. The amnion covering the umbilical cord. Umbilical cord at the top. This is the amniotic surface of the placenta, so you can see those umbilical arteries and veins just under the amnion. And again, in a few minutes, we'll look at how the amnion is formed. Maternal surface. Like a nice beach hat. When my mom worked as labor and delivery, they had a big chest freezer about 4 feet by 8 feet, and they would just throw all the placentas in there, and they had a processing company that would buy them for the protein content, My research organ tissue was placenta when I was at UC Davis, and one of my colleagues, also working on her doctorate, had a bottle of shampoo in the window labeled placenta, and that was one of the high content of protein. I think I'd have a psychological problem washing my hair with placentas, but anyway. This was from a little girl born on Valentine's Day, but I think it was Photoshopped. I mean, there are different shaped placentas. There could be banded placentas where the umbilical cord and strands of fibrous tissue can be wrapped across it. So, anyway, on our models, before we move on to the embryonic disc formation, we have this box that has some of the same models that we have on the drawer, and obviously some glue has come off, unless he's just looking for an A. So we have this model over on the other side too. This is just of a small ovary. So remember, this was the ovulated secondary oocyte with the zona pellucida being white and the follicular cells surrounding it. You can't see the first polar body. And then here we have the secondary oocyte in the process of forming the ootids with the sperm, having just fertilized it, starts to divide. Here we have the polar body is hidden here. We have two polar bodies. Here's the male pronucleus and the female coming together, starting the first mitotic division. So this would be the zygote, which is what we call the fertilized egg associated with the sperm. And then we start going through some of the stages of placentation. Down here would be the placenta. It's about 8 inches across, 7 to 8 inches. The word placenta means cake. Maybe that's why they ate it. Or they ate it and then called it cake. I don't know. What you're seeing here is the chorion. This would be the amnion covering that. And then just a view of the cross-section through that with the amnion and the villi. This would be the decidua basalis down here within the maternal lacunae. We also have somewhere. Here we go. These models. So I showed this one maybe on Thursday. It's what we talked about. So in the direction of implantation shown in the video, moving up into the maternal tissue, the way I diagrammed it, moving down. It's just easier to draw it that way. So this would be the stratum functionalis, forming the decidua basalis. And the dark red would be the trophoblast cells that have fused together. So you would have the syncytial trophoblast, and the cellular part of those trophoblasts now are called the cytotrophoblast. Right? And then this is the inner cell mass having just formed its first cavitation that we're going to talk about. There's another model intermediate to this. Don't see it around. Shows essentially the same thing. Actually, it's not intermediate to this. It's later than this. Oh, I'll do this one. So here we have, we're going to talk about the amniotic sac and the yolk sac that are formed. The extra embryonic coelom, which is the gap between them and the developing placental tissue. The white here would be the cytotrophoblast. The red again, the syncytial trophoblast, maybe it's gray. And then around here would be the maternal lacunae. So all of this is forming the chorionic villi. All right? And we have it starting to bulge up out of its space. And anybody see another model that looks almost identical to this? Yeah, that's what I'm looking for. Thank you, Captain. So this is just a little bit later. Thank you. And so here we have, now you can see distinctly the chorion villi with the villi branching, all right? The maternal lacunae. So now down here we would have the decidua basalis. It would be much thicker. We just start seeing all of it. This would be the chorion levi. You can see how the villi are not very well branched. So that would be the smooth chorion. Above that would be the decidua capsularis, and out here would be the uterine lumen, So the only little bit of decidual parietalus that you might see is out here, where the wall is starting to form a continuation of that lumen. Okay? So those are you can see the amniotic sac here as well, but we haven't talked about that structure. Okay? So being able to identify in lab, or the lab quiz or lab exam, those structures that we've gone over and over. So the original ball, the blastula, its cells are the trophoblast, and resting inside that, the inner cell mass, which is what we're going to talk about in a few minutes. All right? Then the trophoblast cells start to multiply and fuse together. So when we see them fuse together, that's our syncytial trophoblast, and the cellular component, the cytotrophoblast. Then the syncytial trophoblast grows the chorionic villi filled with cytotrophoblast cells forming the connective tissue, and we have chorionic villi versus the chorionic levy. And the maternal tissue, the stratum functionalis, we have the 3 areas of decidua. Okay, so those are the basic structures for identification in lab aspects. Now let's look at the inner cell mass. We're going to go through those same images that I showed you for implantation, ignoring the trophoblast cells and paying attention to the inner cell mass. We will not go into all the aspects of fetal development. We're going to stop with the formation of the complete embryo. So you have a chapter in your text that takes you through all of those, when the eyelids form and open and then fuse again and then separate. But we're going to stick with just the trilaminar disc and its folding, and the formation of the 3 layers of embryonic tissue. So up here, we have our inner cell mass. And as you can see in this image, as the blastula is starting to settle into the stratum functionalis, the inner cell mass undergoes its own changes. So let's look at those. So we have our trophoblast. And cluster of cells resting on the inside of the blastular cavity. Forming a mass known as the inner cell mass. Each of these cells has the same chromosomes as any of the cells forming the trophoblast. All right? Same chromosomes, but different genes expressed. So each of these is still a 2N. Then there is a separation, and a layer of the inner cell mass stays associated with the trophoblast, and a cavitation is formed. So this first layer that's staying associated with our trophoblast, which now is becoming visible as a syncytial trophoblast, and is going to develop into our placenta. This first layer becomes the amnion. We now form a bilaminar or a 2-layer disc. We can't quite yet assign these to terms of embryonic tissue, but the layer closest to the amnion is identified as the epiblast. It's going to give rise to what we identify later as the ectoderm, which is going to form the nervous system, and skin. This layer is identified as the hypoblast. It kind of will form the endoderm, but not as directly. The ectoderm is going to give rise to many of the cells that end up being the endoderm, but they get pushed aside. So they're in the place of where the endoderm is going to be, even if genetically you can't follow it along in the same place. So I'm going to write ... endoderm sort of. And explain it later, all right? Now, this amnion forms a balloon-like cover. It's like I was flat, you know, I'm like a lizard on the ground, and I'm carrying a balloon on my back. That's the way the amnion is associated with our embryonic disc. We have a flat oval disc with a balloon on its back. Now, the hypoblast cells contribute to a balloon on its tummy. We use scientific terms. And we see the yolk sac developing. So obviously, we stay inside our mothers and use their blood supply for nutrients as we develop. We're not born with an eggshell around us, and we are dependent on the nutrients within the yolk for all the rest of our growth material. So the yolk sac isn't a functionally nutritive supportive structure. It does give rise to the blood cells that will migrate along the yolk sac and into the embryo. All right, so it does give rise to some components, although the yolk itself is not an important aspect of it. So here you can see the epiblast, the hypoblast, the amnion with the amniotic sac, and the yolk sac. Now, interestingly, kind of a conundrum, the amniotic sac there is really little, but it develops to encompass the entire fetus. All 6, 7, 8, 9, 10, 11, 12, 13, 14 pounds of it, whatever it weighs when it's born. The yolk sac disappears. All right, so we have a different rate of growth and development for these structures. So here's what I mean by the balloon. So here's what we have with the epiblast and hypoblast. The blue is the amniotic sac. So it grows right off the edges of the embryonic disc. Here's our two-layered embryonic disc with the epiblast and the hypoblast. And those cells change. Here are their epiblast, here are their amniotic membrane. So there's a connection. You ever notice sometimes you can buy cheap toys that you have to snap off of a plastic? Sometimes they come in Cracker Jacks or cereal boxes, and you snap them off. They're like stamped in plastic, and you snapped off the pieces and then put them together. Or maybe they were made by just swirling liquid plastic inside a mold, and you could always see a seam line where the mold came together. So it's a wonder we don't see this seam line where our amniotic membrane used to be right along the side here. And the reason we don't is because this actually ends up being in the middle of your body. Remember the linea alba between the two rectus abdominis muscles? That used to be out here as a flat disc. And as we'll see in a few minutes, there's a longitudinal folding that occurs. So that this flat disc that was bilayered, okay, ends up being trilayered, but it folds around and forms a fusus in the center and forms a cylinder. Now, if I have I'm not tearing the embryo, don't worry. I've never done this before, so we'll have to see how this works. Here's our bilaminar disc. Here's the amniotic sac. Should make it blue like the dorsal body, okay? So now as our amniotic sac curls around and forms a longitudinal folding, look what happens to the amniotic sac. It curves around with it, okay? So here's our flat disc, and then it curves. So inside of that curve I'm forming would be your ventral cavity, and notice that the amniotic sac is curving around with it. So essentially we should have it attached to us right here, all the way down through, and then it's free around. But then you also curve this direction to form the C shape of the embryo, fetal curvature. And as you do so, that amnion is pulled down and only is found around the umbilical cord.
That's crazy.
So it's fascinating. Yeah, it really is fascinating how we go through all those changes. So this would be the disc, and then we have a third layer develops, the mesoderm. So this is showing the conversion. So we go from 2 layers to 3 layers. So if we look at a cross-section of that, cells will move in this direction and migrate down into this groove. You can see here the primitive streak, and that will progress forward to continually have this migration. And these cells, we have our hypoblast here. And if you look at it, it appears that these cells migrate into, and all that happens is they become the mesoderm, which they do. But they also continue to migrate and displace and replace these cells, which are becoming the hypoblast. The endoderm. So really the endoderm comes primarily from the ectoderm. I won't test you specifically on that because it's kind of vague and some texts don't explain it very well. And I didn't actually realize this much of it until I taught the repo physiology class at Sex State a couple years ago. But I just want you to recognize it's not just the mesoderm that the ectoderm forms. So now we have 3 layers of tissue. We have the ectoderm, as I said, becoming the skin and neural tissues. We have the mesoderm, which is going to become connective tissue. So it's going to form the smooth muscle and skeletal muscle and bones and collagen, fibrous connective tissue, cartilage, blood cells from outside the yolk sac. And then we have the endoderm, which essentially forms the lining cells of our tubular structure, so the cells lining your mouth and your stomach and your small intestine, cells lining your respiratory tract, all right, many of the cells forming your urinary system.